Hello and welcome to another episode of the Atherosclerosis Talk podcast, the podcast series hosted by the ES Young Fellows. So I'm Emiel van der Vorst from the Department of Internal Medicine 1 in Cardiology at the University Hospital in Aachen in Germany. And I'm very happy to host today's episode. So we're joined by Professor Noam Zelcer, a globally respected expert in the field of molecular regulation of lipid metabolism and cardiometabolic diseases. Professor Zelcer completed his PhD at the Netherlands Cancer Institute and the University of Amsterdam in 2003, followed by a postdoctoral fellowship at UCLA. And since 2009, he has had his own lab, and in 2016 he was appointed as full professor at the Department of Medical Biochemistry at the Academic Medical Center, AMC, of the University of Amsterdam. So the lab of Professor Zelcer aims to uncover the fundamental mechanisms underlying the molecular regulation of lipid metabolism in health and disease, by studying this in the context of cardiometabolic diseases using genetic, biochemical, cellular, and animal models. Professor Zelcer, it's really a true pleasure to have you with us today. On behalf of the ES Young Fellows, thank you for joining. I gave our audience a quick glimpse of your background, but tell us, is there anything you would like to add to capture who you are?
Thank you, Emiel, for the nice introduction. And I feel very, I think you did it very well. I think I feel well introduced. Thank you.
Great. So then we directly dive into the questions, and maybe the first question that many of our listeners can relate to. Could you maybe describe a typical day in your life, if such a day exists, and how you manage to juggle different tasks like research, teaching, leadership duties?
Okay, well, a typical day is always hard to define, but unlike many, I am very much a night person. So my typical day usually starts later than regularly. So I know there are many people who like to start early. I am, for one, you should not talk to me usually before 10 a.m. So no meetings before that. And I try to avoid anything earlier than 10 a.m. in the morning if I can. I do like to work at night. I find it very peaceful and quiet. So my days are a little bit night shifted. So I'm really a night owl. So that's in a typical day would be usually, as you asked about, juggling the different responsibilities I have, which I think as a leader of a research group is the primary one, is really mentoring my students, my coworkers, if it's on a personal level, but also at the scientific level. So that takes most of my time to trying to monitor the progress, monitor experiments with them together. So that would be the predominant time. And then obviously I have some responsibilities within the institute in which you juggle, you put everything. And teaching, I do a little bit, not that much. And when I teach, then, well, obviously that gets priority for those few weeks in the year that I have to do that, which is also fun.
Great, thanks a lot. So you're currently also on the organizing committee of the European Lipoprotein Club, which is also supported by the ES. Could you tell us maybe a bit more about this club and also about your yearly meeting?
Well, first of all, I'm very grateful that the ES is supporting the ELC. The ELC it's a yearly meeting, the European Lipoprotein Club. It's actually been going on for many, many years. We're coming up on our 50th anniversary soon. It's a wonderful meeting because I feel very strongly about it. It's a small meeting. We're a max of 100, 110 people. It's extremely interactive, and especially for younger people, for trainees, it's a great place to come join and discuss anything related to lipoprotein, lipid, metabolism. And it's a wonderful place to present your work. It's very safe, very pleasant, and it's also a wonderful place to get acquainted to what other peoples within this community in Europe are doing, and to establish collaboration. So I've made many friends, but also many collaborators during these meetings to this date. So this is the one meeting I don't miss any year.
So that's also for our listeners. If you don't know about it, have a look and register.
I should market it. Please have a look. It's usually around September, mid-September. It's always in the same place in Tutzing, so just south of Munich. And it's a wonderful meeting, and the EAS advertises it, and ELC advertises it, and please join us. We always are looking for new groups, teams that are interested in this field.
So we already talked a bit about managing multiple responsibilities. Do you have maybe any advice for also young people that's starting to start with their own group? How do you learn, or how do you train to navigate through all of these responsibilities?
Yeah, it's an interesting question, because all of us that are making this transition, when I made it, but also the younger people making this transition from becoming a full-time researcher to a group leader or some research responsibility, we're actually not trained for this. We're trained to do science, we're trained to do experiments, to interpret it, to publish, right? And suddenly you get many new roles that you are thrown at you, and you're expected to be able to do them. And most of us do that mainly by imitating what we've seen, models we've seen. And some of these models are good, some of these models are bad, and some of these models are actually not suitable for you personally. So I think the tips I would give people, and why I try to help, is I guess one, if you can find somebody to mentor you in the process, somebody you feel safe with and can be open with and can be share the difficulties also. I think that's very important, both scientifically, but also personally as you transition. It can be somebody in your institute, or can be an external coach, which I've had as well. That's one. I think the other thing is to learn very quickly to say no, because especially as young people, you'll be asked to do many, many things, and some are important, some you may perceive as being important, but they are not important. So learn to say no and protect the time you want. It's very important to be part of the institute and the department and contribute to the general tasks, but learn to protect yourself. And the last, I think, which is maybe the most important, is the initial recruitment of your people is so critical. Think well and prepare yourself for this, and you can learn from either reading books, talking with people, because there are many people are selected. You do the first selections based on some perceived feeling, gut feeling, and that's not a good idea to follow. And then the tip would be there, if you interview somebody and you considering selecting, if you have any if you have a question about that candidate, then there's no question. Don't take him or her. Sorry. Yeah. So the selection of people is really critical. These are your make or break decisions, especially as a starting career in science. So take them very seriously. You worked hard to get to where you are, then make sure that you give yourself the chance to actually develop further.
Great. Thanks out for these insights. So you already mentioned a bit about mentors in your past. Can you maybe tell a bit more about this? Which mentors did you have, and how did they shape your career or your journey?
So I had two people which I see as mentors. One was my PhD supervisor, Professor Piet Vorst at the NKI. He's a very famous scientist in the Netherlands. And from him, I really learned to think critical, do critical science and think critically about the results and what they mean, and not just do experiments for the sake of experiments, but think. That's a word he used a lot. And the other thing I learned from him, which I take to this date, is scientific integrity and the importance of being honest about your research. And this is something that is very important for me personally, and I'm also a member of the University of Amsterdam Scientific Committee, Integrity Committee. So it's something I really, I think it's an important thing to emphasize. And I still, once in a while, when I have dilemmas in science, I think, what would we do? So I think that's a good thing. And the second mentor would be my postdoc mentor, which was Professor Peter Tontanos at UCLA. And from him, I really learned, one of the things I really learned from him a lot is to write. He's a fantastic writer of scientific writing, and he's been a great teacher in that respect. And also from him, how to package a set of experiments like into a really strong article. These are still lessons I follow to this date whenever I can.
Before we go into the hardcore science, maybe also more on a personal note, do you have any hobbies or activities outside of research that can help you unwind and think about other things?
Well, the first one is kind of, I don't know if it's embarrassing or not, but it's a sort of a boomer type of a thing. I love gardening. I love plants. I love working in the garden, and I love to see the seasons working through the garden, and I enjoy that a lot. It's very peaceful. Also, especially because plants do not speak back to you, so it's very, very quiet. The other thing, I took on piano playing a few years ago, which has always been my dream. Well, it's going slow, but it's going. It's a shame I didn't start earlier, but it's always been my dream to do it. So I'm getting better at it as I go. And the last thing, I used to be a professional handball player. Due to injuries now these days, I've injured my ankle, so it's a little bit hard for me to run, for example. But I try to sport as much as I can, and wherever I can get opportunities, I do that. So it's a combination. But it is very important to have hobbies, to have a life out of science.
It's also great to have a very diverse set of hobbies.
And it's important to have good relationship, friends. These are all things that will support you as you go through your scientific career.
So let's move on to some more scientific words. So you focus a lot on lipids, cardiovascular disease, metabolism. But what originally drew you towards this scientific niche that you have? Were there specific moments that really shaped this path?
Yeah, that's always tricky. I'm not a person who did start my training and thought, this is what I want to do. So like I think many things in science, a lot of serendipity. The science leads you to where you are. So when I moved to do my postdoc in UCLA, I knew I wanted to work on something which is related to metabolism. And then I went to a lab that was very focused on cholesterol metabolism. And I actually started looking at cholesterol metabolism in the brain, in Alzheimer's disease. And through a very, at that time, microRNA experiment, the equivalent nowadays of an RNA sequencing experiment, I found this new gene called IDOL. And that really, which maybe we can discuss, it's a regulator of the LDL receptor, which really shifted my research more towards the lipid, metabolism, cardiovascular niche. And so it's really that particular finding that really took me this direction. And then I've been obviously developing it further.
Very interesting already. So also of course, the field itself of lipid metabolism changed over the years. Can you elaborate a bit how does this also affect your work that you're doing in your group?
Well, yes, it has developed a lot, and we understand a lot more, and especially also in terms of therapies. I think we have a lot more therapies nowadays to treat all sorts of dyslipidemias. Not all effectively yet, but for many, yes. I think where there's still a very big gap is trying to understand the regulation of lipid metabolism at the molecular level, at the cell level. So to really understand the fundamental issues that at the end govern if you have more LDL, less LDL, or LPA more or less. So I think that's where I try to focus my research on mainly these days, and I think many things that are helping is really the technology. So we can do lipidomics nowadays. We can do genetic screens, which my group does a lot. We can do a proteomics, very advanced proteomics these days. So I think that has been really propelling the very fundamental understanding of how lipid metabolism is actually regulated. Well, at least that's the direction that my group has taken. Yeah.
So you already mentioned that you're looking into molecular pathways and also mechanistic work. Is there any particular mechanism or molecular pathway that you think is currently underappreciated, or and maybe also has the potential to go further towards some therapeutic approach?
Yeah. So I do think I was thinking about this question for a long time, and I think there are two. When I think two things I want to get into, one of them we're already looking into a little bit. Three actually things I think I would like to mention. First, I think is the lipoprotein uptake that is not LDL receptor dependent. I think this is there are now more and more examples with the angioprotein like 3, for example, that there's clearly a way to clear LDL, which is LDL receptor independent. And how that works, I find fascinating, and I'm very curious to try to help with that. Another thing I'm very interested in is in the whole process of how is VLDL actually produced in the liver. So obviously we know it's made in the liver, is secreted, but the whole process of trafficking a very large particle through the secretory pathway is not trivial. The lipidation process of VLDL is actually also not completely resolved. And so this thing we're actually actively now involved. And the third thing, which is much newer in my group, we're trying to develop is the idea of RNA editing, which is getting traction these days, just like CRISPR, like the CRISPR revolution. This is a whole new field that is coming up, and I think there's many opportunities there for therapy. And I think on all these three, if we understand them better, there's way to therapeutically engage these pathways, and these technologies. So we'll see. We'll see where the future will bring us.
And great. So you already said yourself that let's see what the future will bring. Maybe from a more broader view of the cardiometabolic field, do you think are we moving more towards like approaches to combine really lipid, immune, metabolic systems?
Well, especially now we're recording this during the EAS in Glasgow, and if you looked at the talks, it's clear that there's a broader shift from just only lipid lowering or lipid targeting to inflammation and other risk factors, which are, you would think. I think they are not mutually obviously exclusive. I think lipid metabolism is directly related to inflammation in many cell types, so there's direct links. So I think, yes, the field will expand to integrating different elements of the cardiovascular disease repertoire from different directions. Sure, that's the future.
Before we go towards the ending, we always also include a reflective section we called the graveyard of ideas. So we believe, as also young fellows, that failure is a natural and maybe even essential part of science. And even if you don't talk enough about this. So could you maybe share an example of a research idea or a grant or a project that did not go as planned, and maybe did you learn a valuable lesson about this?
So first of all, I encourage you all to discuss your failures, because when you share them with other people, you'll see that many people have stories to share as well. So you'll feel better, but also you'll learn how to deal with them better with each other. That's one. Two, I think it's failures are a feature of doing science. They're not a bug, actually. So it's inherent to doing science. So one, for example, is that still hurts me, is we've been studying an E3 ligase called RNF 145, and we basically spent a lot of time on it, got stuck, and at a certain point we pretty much dropped it. And that's a big failure, because I had great expectation for that project, and I think with hindsight we should have approached it differently, but hindsight is a very bad agent. And I think one lesson I think about failures is that I think all of us have the tendency to push projects and go on, even if we start running into very large walls, and then you say, okay, but I've already invested 2 years in this, well, I'm not going to stop now. And then a year later, you're still not much further. So I think one thing we can learn from pharma industry, for example, is to be very strict with no-go steps in our research. Sometimes it's better to stop something after a few months than losing another 2 years later and realizing I didn't get much further. I should have, because there's always something else to study. There's always things to study and discover.
Yeah, I think there's already a great advice also for the young people to keep in mind also for their projects. So maybe one last practical question also for our listeners. Do you have any open vacancies in your group at the moment, or do you advertise them?
This is a fantastic question, because as we speak, I actually do have an open. It's a PhD position for a zone. So the Dutch scientific organization funded project to study cholesterol handling in lysosomes and different cardiometabolic conditions. So it's a PhD position at this point. I advertise it, so we have a standard procedure, and I recommend, so we do it through we have the institutional website, which probably everybody has. In the Netherlands, we also have like a central website where all academic positions are listed. It's called academictransfer.nl. So it also shows up there. And then I use LinkedIn and my whole network to share it. And there's also another tip again. There's also a European, EU website where you can post jobs for free. I don't have the exact URL in my head, but if you look for it, you'll find it. So usually that works. And the best candidates you get will be those from your network. So have a big network.
That sounds great. So also for everybody who's listening, who's interested, look on to the websites, reach out.
www.zelcerlab.eu. Please, it's listed there as well.
So this has really been a very insightful conversation. Professor Zelcer, thank you very much for sharing your time, your experience, and your perspective with us today. So to all of our listeners, thank you for tuning in to EduTalk. If you enjoyed this episode, please consider subscribing, leaving a review, and sharing the podcast also with your colleagues. So we love hearing your feedback and suggestions, so don't hesitate to let us know what topics you would like to discuss, also in next episodes. So until next time, stay curious, stay inspired, and thank you for listening.